Hey everyone, Moms Against Medical Bullying. So I'm going to continue reading this article here. The next section is called DNA Fingerprinting Principles and Forensic Applications. The first technique of this type was developed in England by geneticist Alec Jeffreys in the mid-1980s, who was looking at techniques to screen for hereditary diseases and genetic defects. However, when asked by the local constabulary to investigate a rape case, he successfully identified the rapist from his DNA profile. Within a few years, DNA profiling-based techniques were used by forensic scientists around the world. Unique DNA sequences are present in all species and are used as biomarkers for the detection of cells from that species. These DNA sequences can most easily be detected using the polymerase chain reaction-based DNA fingerprinting methods. All DNA fingerprinting techniques produce a pattern or profile of nucleic acids, which are amplified by PCR, from a sample, and that pattern reflects the microbial community structure from an environment. DNA fingerprinting or profiling comprises any DNA-based techniques that identify the DNA from a certain individual or a group of individuals within a community of organisms. DNA fingerprints may be used as a tool for determining the identity of a specific DNA sample or to assess the relatedness between samples. Most DNA fingerprinting methods are highly specific, highly sensitive, and largely independent on the physiological or growth state of the organisms, which makes them of particular interest for forensic investigations. The principle of these techniques consists of screening for certain DNA sequences that are found in some individuals but not in others, thus visualizing DNA polymorphisms between samples and producing specific DNA fingerprints. Each individual has a DNA profile as unique as a fingerprint. In the case of humans, for instance, over 99% of all nucleotides are identical among all individuals. However, for every 1,000 nucleotides inherited, there is one site of variation called polymorphism in the population. These DNA polymorphisms induce the change in the length of the DNA fragments produced by digestion with restriction enzymes in the course of fingerprinting technique. So DNA polymorphism determines the resulting fragments to be of different length and or sequence. Gel electrophoresis is used to separate and determine the size of the resulted fragments. This is because DNA is a charged molecule. That's interesting. So in an applied electric field, it moves toward an electrode. The exact number, wow, that's so interesting. Gel electrophoresis, used to separate and determine the size of resulted fragments. DNA is a charged molecule, so it moves toward an electrode. The exact number and size of fragments produced by a specific restriction enzyme digestion varies between individuals. The fragments are arranged in order of length and tagged with radioactive probes emitting X-rays. So when the sample is photographed, they show up. This produces the fingerprint represented by a series of black lines corresponding to the DNA sequences present. Many DNA fingerprinting methods are available. DNA fingerprinting techniques with potential applicability in environmental forensics are listed. They differ methodologically, using various techniques to look into the polymorphism of certain genes. All these techniques function based on the same principle described earlier, and they all result in producing specific patterns of microbial communities in the environment. DNA fingerprinting has been increasingly applied to determine the ancestry of plants, animals, and other microorganisms. Another application relates to tracking genomic mutations, molecular diagnosis of complex inherited disorders, population screening of genetic diseases. This is interesting. Studies on the genetic basis of variable drug response, as well as discovery and investigation of new drug targets. Involving screening for mutations in multiple DNA samples are only a few examples of DNA fingerprinting application pool. It just makes me wonder, like, and kind of realize that this is, I feel like this is a main reason that they want everybody in this healthcare system, because they want to collect all this information about us. They want to know about our genetics and our gene pools and stuff like that. And this section also made me realize, in a way, because somebody I know would say, what if they release specific vaccines for specific areas or whatever? And I thought, I didn't really give it much merit, but then with realizing that they do like to keep track of our genetics, hence, wanting people to go to ancestry.com and, see what your genetics are. Well, they want to track our genetics for their studies and stuff. So I guess something like that would be possible if they collect information on us in, all over the world, all over this, everywhere, what is going on in specific areas of the world. But it's pretty interesting. This is, I don't know, it kind of makes sense that this is why there's such a huge monopoly. All right, forensic applications. Microorganisms respond to contamination. This is interesting too. And such response may be recorded by DNA fingerprinting methods. The response to heavy metal contamination was well studied and could be the basis for forensic applications of microorganisms. Biological processes are particularly sensitive to soil heavy metal loadings. The negative impact of heavy metals results from their toxicity to biological processes. The population size of rhizobia has been found to decrease as a result of metal contamination. Several studies that use plate count techniques have demonstrated an increase in gram-negative bacteria and a shift in the composition of fungal species toward a more metal-tolerant community in metal-contaminated soils. Therefore, it is postulated that the soil microbial community could serve as an indicator of losses in soil quality due to heavy metal contamination. Such indications may provide the basis for developing innovative forensic techniques for tracking metal contamination passage through the environment and identifying the source of contamination. So that's really interesting what they're saying there about using microbes to determine, they can determine what kind of contamination is happening due to what microbe is present. So that's really interesting. Maybe that's what they're doing with us, They make changes to the environment and then see how it affects the microbial community inside of us. So anyway, it's just something to think about.